Good morning, I'm Beatrice U Laquian, and together with my partner,
Gillian Reyes, and we will be discussing our study, which is the stacking efficiency of modular-sediment microbial fuel cells deployed on polluted river sediment.
So to give you a short introduction about sedimentary microbial fuel cells, or SMFCs, first of all, it is a bioelectrochemical system that converts organic matter present in the sediments to electricity while acting as a wastewater treatment system. This happens when microbes in anoxic conditions break down organic matter to produce electrons, and this cycle continuously reduces organic matter. SMFCs are a relatively new topic. They are known to have lower energy outputs, but studies have shown that several factors, such as microbe species, electrode material, substrate, and system design may help to increase it. This study focuses on the last one, which is the system design, more specifically the configuration and stacking applications of SMFC systems. We chose this because there are no existing studies that optimize in situ applications of studies specifically in the Pasig River. Stacking SMFCs have seemed to amplify the power output, but the number of cells and their configuration may greatly affect the output. For our objectives, we aim to design a stackable SMFC system that can be deployed in situ, to determine the optimal configuration for stacking applications using the data on voltage output, and finally to determine the removal efficiency of chemical oxygen demand and the total nitrogen.
Methodology. Here is a brief overview of our research. First is the collection of sample from the Pasig River. However, due to the pandemic, we weren't able to obtain our sample from the Pasig River itself. Hence, we settled with its tributary, which is the Pateros River. Then, the samples were sent to fast laboratories to obtain our initial values for COD and total nitrogen. Next is the preparation and gathering of materials for our SMFC system. The cells were then constructed and deployed in our polluted river sample. Then, the daily measurements of voltage and current for individual cells were done. Next is the daily measurement of voltage and current for 3-cell and 9-cell series and parallel connections. Next is the daily measurement of voltage and current for cumulative stacking for both series and parallel configurations. Next is the daily measurement of voltage and current for hybrid configurations. Class C for measurements is the polarization measurements of individual and different configurations for every 5 days. The samples were then again sent to fast laboratories to obtain its final COD and TN values. Lastly is the treatment of data and analysis of results. Sample testing. The samples were collected and sent to fast laboratories in order to determine the COD and TN values. The test method conducted for COD was closed reflux titrimetry, which utilized a setup of fabricated 6-placer equipment. On the other hand, the Jadal method used on the determination of TN utilized Geltec for its digester, and a Gerard Vapotest was used for the distillation. Sediment microbial fuel cell design. All 9 cells were constructed using polyvinyl chloride pipes. Then, for the electrode, carbon fiber was used for both anode and cathode. As stated before, the sediment sample was obtained from the tributary of Pasig River, which is the Pateros River. Lastly, a non-woven polyester cloth was placed in the middle of the cell in order to ensure that no sediment mixes with its river water. Here is our SMFC setup. As can be seen in the figure, all cells were connected in a 3x3 connection. The anode, which is present in the bottom part of the cell, is buried deep beneath the sediment, while the cathode present at the top of the cell is present in overlying water. There are 2 main configurations for this experiment. First is the series, and the second is the parallel configuration. For cumulative stacking, 1 cell is added until all cells are connected in either series or parallel arrangement. For hybrid configurations, there are also 2 main types. First is that 3 cells that are connected in series are then connected in parallel, and the other one is that 3 cells connected in parallel are then connected in series. For polarization, first, the external resistance is varied from 30 to 50,000 ohms. Next, the voltage is measured and the current is calculated. Lastly, the power and current density were calculated to generate the polarization curves. Here is the actual setup. Samples were sent to the FAST laboratories to obtain COD and TN values. Next is the interior of the cells, showing the electrodes and the non-woven polyester cloth. 9 cells were submerged in the sample and the gathering of the cathodes and anodes together with an external resistance.
Results and discussions. As shown in the figure, the parallel configuration ultimately delivered the greatest voltage output. The decreasing trend shown the standard deviation of the sample. This is caused by the degradation of the organic matter present. This shows that the SMFC is working as a wastewater device. Now we have the voltage generation by SMFC in a series configuration and a parallel configuration. In theory, series configuration should have produced the greater voltage output. However, this was not reflected in the data. This is a common problem for microbial fuel cells in general and not just SMFCs. Series configurations are more susceptible to electrode reversal or voltage reversal. Voltage reversal is the drop in the voltage that occurs when the voltage of the anode is less positive than that of the cathode. Because of this, parallel configurations are more currently favored. The causes for voltage reversals are bacterial starvation, substrate concentration gradient between the cells, blockages, impedance difference, and faulty cathodes and anodes. Now we have figure 3, which is the current generation of an SMFC system for series and parallel configurations. When looking at the data for the current generation, the parallel configuration is still favored as its results were closer to the theoretical value. The percent error for series connection was 34.75% for with and external resistance and 33.81% for without, while for parallel it was 3.55% and 5.48% respectively.
Figure 4 is the cumulative stacking for both series and parallel configuration. This compares the actual and theoretical voltage for when cells are added one by one. This helps determine the optimum number of cells and whether there is potential loss. For series, it can be observed that the actual only intersects with the theoretical when the number of cells is only equal to 1. This may mean that there is a huge potential loss suffered when the cells are connected in series. On the other hand, for parallel configuration, it can be seen that the actual lies much higher than the theoretical, which may mean that there is no potential loss suffered when the cells are connected in parallel. Figure 5 shows the current and voltage generation by cells in series parallel configuration and parallel series configuration, with external resistance and without external resistance. Theoretically, in parallel configuration, the current increases while its voltage remains constant. On the other hand, for a series configuration, it is typically observed with voltage reversal, and in hopes of negating it, hybrid configurations is done. As observed in the figures, the current and voltage was much more higher when the cells were connected series and parallel configuration. Figure 6 shows the polarization curves showing the current density versus power density and the current density versus power density and voltage. Figure 6a shows that the greatest power density is at the first day of the measurement of polarization, which is at day 5. This was observed for all configurations. However, the lowest power density was generated when the cells were connected in series configuration, which was caused by voltage reversal. For figure 6b, considering the voltage measured for all configurations increased as the external resistance was increased, it would mean that substrate starvation was not a factor for the decrease in figures in 6b, since voltage starvation is prone in higher external resistances. The slope of the voltage decreased and was almost linear, which may indicate the dominance of ohmic losses. For the current, considering that the current decreased as the external resistance increased, it is consistent with the literature and showed a typical behavior of a microbial fuel cell.
Now we have Table 1, which shows the final and initial values of the COD and total nitrogen as well as their percent removals. As shown in the data, the SMFC system design was effective as a wastewater device. This is reflected in the high percent removal of both COD of the water and total nitrogen in the sediments. This can further be proven by looking at the average COD of previous studies. The average of an SMFC system was 91.51%, and for a microbial fuel cell, it was 74.51%. A closer approximation would be constructed wetlands microbial fuel cells, which only had an average COD removal of 79%. However, it was found in previous studies that this percent rose to 98.5% when stacking is applied. As for the total nitrogen removal, the result of 96.77% is optimal since the average for any SMFC system is around 63.21 to 96.54%. Since the nitrate and nitrite denitrification occurs in the cathode, the high percent removal means that the cathode surface area was optimal in this experiment. Now for our conclusions, we conclude that the highest voltage generated by individual, series, and parallel stacking are 90.8 mV, 63.8 mV, and 255.1 mV respectively. For hybrid configurations, 3 series in parallel exhibited better performance than 3 parallel in series arrangement. The highest power density generated by individual, series, and parallel stacking are 1.83 mV per meter squared, 0.06 mV per meter squared, and 4.25 mV per meter squared respectively. The maximum power density is achieved by 3-cell pure parallel stacking having a value of 12.80 mV per meter squared. Both COD of water and total nitrogen in the sediments were greatly reduced, having a removal efficiency of 98.9% and 96.8% respectively. As for our recommendations, we recommend looking into ways to limit voltage reversal to increase the voltage output, to consider the significance of temperature and pH to improve power generation, to consider the COD and TN as parameters in determining the optimal configuration, and to perform the experiment on-site.